Stomata. Distribution of stomata in plants. Mostly confined to the lower surface of the plants, dicotyledons. Grasses have stomata on both surfaces divided equally, mainly monocotyledons. Floating plants have stomata on their upper surfaces. Submerged plants lack stomata. Pines, gymnosperms, have sunken stomata to prevent water stress. Shapes of stomata. Two dicotyledons have two bean-shaped stomata. 3 monocotyledons have elongated stomata. Histology of stomata. Stomata have lower chloroplast content than the surrounding subsidiary cells. Guard cells have radial myceliation, microfibrils, which prevents them from swelling, i.e., increasing in diameter. The guard cells have thickened on the concave side to favor opening. The guard cells are attached at the top and bottom ends, which prevents the elongation of guard cells. The restriction of movement of guard cells due to swelling due to water causes them to open. This was physically and experimentally proved by Donald E. Ayler, 1975. Stomata opening takes at least 1 hour, but closing is instant to manage the stress. Light is important for the opening of stomata to facilitate photosynthesis as CO2 levels decrease in the mesophyll, causing stomata to open. Like photosynthesis, it opens with blue light only, as has been shown in studies. Blue light causes an influx of K+ ions in the guard cells, the main causative factor of stomata opening. CO2 and ABA levels, Zager, 1977. Stomatal mechanism of opening and closing. The mechanism of stomata opening is illustrated below. There is an increase in turgor pressure in the guard due to the inward and outward movement of water. The pressure values range from -1.9 MPa open to -3.5 MPa closed in. This proves beyond doubt that water is responsible for the opening and closing of stomata. Let us see the factors responsible for water movement. Many environmental factors are responsible for water movement, as follows. 1. Light. It not only opens and closes but also influences the rate and aperture of stomata. The amount of sunlight required for this effect is 1000 to 1/30th of full sunlight. Stomata are open in sunlight. A. Closes at night. B. Close in afternoons. C. If we transfer plants from light to dark and vice versa. The reverse happens in succulent plants. 2. Carbon dioxide levels, both in the cell and outside the cell, influence the opening, closing, and size of the aperture of the stoma. Low CO2 levels in mesophyll cause the opening of stomata. High CO2 levels in the environment caused its partial closure. Extremely closed stomata did not affect levels of CO2 in the environment. 3. Water potential stress has an equal opposite effect on stomata opening. At stress, A. No stomata will open even if other conditions are favorable. 4. A high temperature above 35 degrees Celsius hurts the opening of the stomata. This explains the heat management of plants by transpiration. 5. Wind. High wind speed closes the stomata and opens gradually as the speed is reduced. Factors that affect respiration and photosynthesis have an equal effect on stomata opening. K+ ion movement theory of guard cells. K+ ions move into the guard cells from the surrounding secondary cells. This causes a rise in the concentration of solute in the guard cell, thus favoring osmosis. Potassium, being a strong reactive ion, forms salt with the anion in the guard cells. A value of 0.5 m of K+ ions causes a 2 MPa rise in osmotic pressure. This proves beyond doubt the importance of K+ ions in water movement in guard cells. Outlaw 1982. Due to light, the starch in the guard cell is broken into PEP, phosphoenol pyruvate, which gets converted to malic acid. The malic acid dissociates into malate anion and H plus ions. These H plus ions move out of the guard cell as the K plus ion moves in to keep cells neutral. Stomata opens when the luminescence intensity of light is 2.5% of the total sunlight on a typical sunny day. The value of light varies in plants. Its lowest value is in tobacco plants, Nicotinum tobacco. The luminance value for stomatal opening is lower than the value needed for photosynthesis, i.e., 200 to 1000 mol. M1.sec-1